Hi, I'm Don Pfister, and I'm here at the International Mycological Congress in the Netherlands. It's August 14th, 2024, and I'm here with Ben Wolfe. And Ben is going to tell us about his career and about his background. So,
Yeah.
Ben, I know you because I knew you as a graduate student.
hmm
Uh, Tell us about what you were doing before graduate school and how you kind of came into mycology.
My original interest in biology is plant ecology. I started studying sort of big, obvious things, plants, when I was an undergrad. I was working in wetland ecology, and I was specifically studying in upstate New York. I was studying fens, these calcareous fens, where these beautiful plant communities that are really species rich, really relatively rare. And I was interested in what constrains that diversity and what forms those communities. And for my honors thesis, when I was an undergrad at Cornell, I started studying mycorrhizal fungi as one potential mechanism for what these plants are doing to live in these particular environments, and looking just at that point, just doing some staining to look at mycorrhizal fungi, but eventually started to get into some of the molecular techniques. And what I learned is that the plants were cool, but I was much more excited about the invisible things that we
Right.
don't really know a lot about. And that's how it all began.
Uh-huh.
Yeah.
And you came to graduate school at
I did.
Harvard.
Yeah, I first took a detour to Canada. I went to the University of Guelph and worked with John Chloronimos for 2 years on more mycorrhizal things to get a little bit more familiar with the techniques and tools. And then I met Anne Pringle, and was really excited to work with her when she started her lab at Harvard to more broadly understand mycorrhizal fungi in the context of invasive species. I was really excited about this idea that there could be mutualistic fungi or symbiotic fungi that could be moved around the world and become invasive, just like invasive plants.
So what was the system that you were working on then?
Yeah, so that was the iconic Amanita or Amanita, depending on how you want to say it. And I was really interested in the whole genus for some of my questions for my dissertation, specifically about how did those fungi evolve to be symbiotic? What were the evolutionary transitions? What were the ways that the genome changed? And then another particular thing that I worked on is thinking about this invasive Amanita phalloides, and understanding where is it in the world, how is it moving around, and what are some of the impacts of that fungus. Yeah, I spent a lot of time chasing Amanita as much as one chases mushrooms. Yeah.
Well, I'm interested in what when you're out in the field, when you were out in the field, what was one of those times where you thought, oh my God, I really did it?
Yeah, there are actually quite a few moments. There was this one particular group of Amanita that are hard to find, but really important for solving this mystery of the evolution of the group. And these are free-living saprotrophic Amanita, which we don't really think of because we think of the group as being ectomycorrhizal. And so I specifically had to go out and try to get them and then culture them. I needed them to be something I could work with in the lab. And so I had a whole network of people in the UK. I had folks in Hawaii. I had folks in Australia and other parts of the world looking for these species so that when they popped up, I could get on a plane, fly over to where it was, grab the mushroom, get it to a lab as quickly as possible and try to grow it. And I think my favorite experience was Jeff Kibbe. We were working in a hedgerow north of London where he knew Amanita anapanada. I'm surprised I remember all this stuff, actually. It's been a while. The unexpected Amanita, which I just love that name. It's been found in New Zealand and the UK, and maybe since I was working on it in other places. And I really needed it in culture. And so he took me to this hedgerow north of London where he knew it always occurred at about the same time. And when we got there, we couldn't find it. We kept looking and looking and looking. He's like, I know it's got to be here. I know it's got to be here. And we were ready to give up. It's sort of a classic thing when you're looking for a particular fungus, ready to give up. We were literally turning around to go to the car, and there was one mushroom. It was a kind of a sad-looking mushroom, but it was all we needed. We took it over to Kew. I was able to get a culture of it, and it was a really important part of my thesis. And so those were great times. I honestly don't do as much field work anymore. And I miss the sort of adventure of going out and trying to search for these elusive fungi that are hard to find sometimes.
But you do different kind of field work
I
now.
do. Maybe the field work is what helped me pivot into a different direction. So for my postdoc, I started working on the fungal communities in fermented foods. And so I loved Amanita, I loved that group, I love working with ectomycorrhizal fungi, but I really wanted to do something that was a bit more applied and potentially had industry connections. And there was a new lab starting at Harvard trying to understand how microbial communities form and function, both bacteria and fungi. This was with Rachel Dutton, and she took me on as sort of the mycology person to help her work on these cheese rind microbial communities. So I learned a lot about penicillium, about a lot of really interesting yeasts, and then that's what my lab has been working on for the last 10 years, ever since that work. Yeah.
So tough life, huh? Jeez.
It's pretty great, actually. Now, instead of having to zip on a plane to get somewhere really quickly to get that mushroom, I just go to cheese caves or cheese shops, and everything's in one room. It's domesticated. It's not natural in any way, but we're still learning a lot of really interesting biology and mycology from those systems. Yeah.
And high diversity?
They're relatively low in diversity. So a typical cheese, depending on the type of cheese, if it's a wash rind cheese like a Limburger, that might have maybe 5, maybe 6 or 7 different fungi and 15 to 20 bacteria. But a typical cheese is very low in diversity, so 2, 3, or 4 species. That's not to say that the fungi aren't interesting. There's a bunch of fungi. We find some Chrysosporium species. There's a whole group of fungi, Sporodonema casei, these orange spots you sometimes see on cheese. And we don't really know much about where they come from and what they're doing in those systems. And so that's some of the work we're doing in my lab, is to try to figure out their natural history. If you go out and look for them in cheeses, are there different strains that are there? How are they evolving? And how do they interact with a lot of the other microbes in the system?
So in a way, it's a kind of mini biodiversity project.
Exactly. We say it's sort of like a little microbial island that people make for us, and we study what ends up growing on that and how they interact and live there. So I am going back to my roots, though. I'm starting a project literally in my own backyard. I've become really fascinated with penicillium in the wild, right? We know so much about penicillium from a secondary metabolite perspective. We know so much about penicillium in food. We know about the ones that spoil food and the sort of bad penicillium. But we don't really know a lot about what your average penicillium strain is doing in soil or on plant leaves or in other habitats. We know what they are, and there's great taxonomy happening there, but what are they actually doing in the systems? And so with Anne Pringle, my PhD advisor, and Cobus Visagi, a penicillium taxonomist, we're starting to go out and sample forests and fields around New England to see what penicillia species are out there, and then more importantly to figure out what are they doing. Are they playing roles in decomposition? Are they mostly just spores that get everywhere and we're just really finding fungi that are good at sporulating and hanging out for a while? And that's kind of fun because it kind of takes me back to my roots as more of a soil ecologist.
Yeah. Interesting.
Yeah.
So any interesting penicillia that have shown up so far?
So the one I love the most, and I'm going to get the name wrong because I just learned from Cobus that actually we're calling it the wrong thing, Penicillium multicolor. So if you take a soil in any part of New England, some parts of the Midwest, and you plate it out, a grassland soil or forest soil, you'll often get these bright orange colonies. So they'll grow up and they'll have this really, it's like almost like a highlighter around the outside edge of it. And the old name for it is Penicillium multicolor, but according to Cobus, it probably should, I don't even remember the name that he just told me a couple minutes ago. It probably should have a different name. Martha Christensen found it back in Wisconsin when she was doing her work back in the 60s and 70s. And Anne and I have been digging up a lot of her old papers in her archives to learn more about what are the penicillium species she found back then, and are we finding similar species today. But that's my favorite just because it's beautiful. It's very striking. The other interesting thing about that species, and a question we're trying to ask, is, I think for many people, if you think of Penicillium or Aspergillus, you think that most species are everywhere. We think of them as very cosmopolitan, but no one's really tested that or really looked
Only
at
with
that.
a few things.
Exactly,
Yeah.
exactly. So Penicillium multicolor, we'll call it, is a great example of one that has what we think is a restricted distribution. So we've been using globalfungi.com, which is this amazing new database that some folks that are at this meeting have been talking about, where they pull together all these high throughput sequencing studies into this database, and you can look up essentially the distribution of fungi through the database. And so we've been using that, but also sampling we've been doing in New England and sampling that Cobus has been doing. And he never finds it in Africa, for example. In South Africa, or we don't see it in any other parts of most of the tropics, but the northern hemisphere, you can find Penicillium multicolor quite commonly. So it might be an example of a species of Penicillium that actually does have a biogeographic distribution. So, yeah.
It's interesting. I don't think I've ever seen it, though I've certainly been in New England and doing soil and other things.
You know, it's pretty easy to grow, and we've found it. So we've sampled 10 sites across Massachusetts so far as part of our preliminary survey, and we found it pretty much at every site. It does seem to maybe prefer more open sort of grassland herbaceous sites compared to forests, but it seems pretty common. Next time you're around, let me know, and I'll bring some over and I'll show it to you, because it's pretty stunning. It's really gorgeous. Yeah.
No, I'm just surprised with having students do isolations in labs over many, many years.
Yeah.
I'm surprised it never turned
Interesting.
up.
We're growing it on a medium called DG18, which is pretty good at just growing mostly Aspergillus and Penicillium. It might have this more orangey appearance on that medium, although my understanding from Martha's papers and others is that it's very characteristically orange around the outside edge. But yeah, I'm going to have an undergrad do a project to try to understand its ecology, so we're going to be sampling it more, hopefully. Yeah.
You know, Martha Christensen was the president of MSA.
I do. I've learned a lot about her. I've been reading her papers, and the thing that I'm so impressed about, since we're talking about sort of history and stories, is when you look at some of her papers, I have such an appreciation for back in the 60s and 70s when people were making figures. There's this beautiful figure where she made an ordination like we do all the time in microbial ecology, but she made it out of wooden pegs. It's like a physical principal components plot where the peg length was an indication of like the abundance of the fungi, and the relative position of the pegs showed the clustering. It's so elegant and so beautiful, and it's been a fun way for me to think like ways that people visualize and thought about data in the past and now. And I've heard from Anne. Anne actually met her. I think a few years before she died, and they had lunch together and talked about some of this kind of work. So it's fun to think about carrying her spirit forward, and in terms of her research and sort of thinking about.
She was a wonderful person.
Yeah.
I think I was secretary in MSA when she was president.
Yeah.
And so, we were back and forth quite a bit. But
and really fundamental work of just where are different microfungi across the landscape, and that data is super useful, I think, for the things we're thinking about now.
Yeah, no, it's great to be able to go back and look at these and realize, she was using techniques and approaches that sometimes we think kind of old-fashioned.
But I love them. I love growing and culturing fungi because you see them, you work with them. I like sequencing data, that's great. But sometimes it removes you from the actual biology, I think. And so I love the work that she's done. And another question we're asking, and Anne's really excited about as well, is with climate change, with increasing temperatures, especially for Aspergillus and Penicillium, the two groups we're thinking about, will the world as it warms become more of an Aspergillus world as opposed to a Penicillium world? Because Aspergillus generally we think of as liking it warmer, Penicillium generally likes it cooler. And so we're beginning to sort of measure that and test that and think about what that could mean for the distributions of these species in the future.
Yeah. Very, very interesting.
Yeah.
Very cool.
It's sort of a fun, again, back to my roots, going back to the soil.
Yeah. Well, you teach now, right?
I do a lot of teaching. It's part of why I like this work, because it's fun to do the science and the research, but it's also fun to pass that knowledge on and get the next generation of fungal biologists and microbiologists excited about what we do.
We should say that you teach at Tufts
Right,
University.
at Tufts University. Yeah, we do a lot of teaching. Yeah.
And what courses do you teach?
So I do a couple. One is an experimental course where we essentially take small projects from the lab into a course setting, but it's independent research, and students learn how to do research through the lens of the projects in the lab. And the last time we did it, it was all about penicillium. And so we did things like genome sequencing. Students did interaction assays with them, and honestly, it was, I think, the best iteration of the course, because fungi are so popular right now, and students are so engaged with fungi that they all fell in love with their individual penicillium and wanted to know more about it. I also teach microbiology, and my goal in microbiology is to get more fungal information into what is generally a bacterial class.
Right.
And again, that's been very well received by students. It's mostly pre-meds, but it's been great.
Yeah.
Yeah.
Good.
Cool.
So, just to end,
Yeah.
what would your advice be to young students?
Wow. Yeah. I always tell people to be curious, but be curious in a productive way. It's challenging. Sometimes you can be curious and just spin around for a while, which I've definitely done in the past. And the way to think about how to be productively curious is to watch how other people ask questions and how they develop projects and how their scientific journey develops, and think about, what are interesting things that you could explore but maybe won't necessarily develop into a full project, versus things that there's a really cool question, there are great tools available. I think that would be what I would say. The other thing is just love what you're doing. Get excited about different. I love cheese. I loved Amanita. I still do. I'm now a big advocate for penicillium and Aspergillus diversity. So fall in love with your organisms, because at the end of the day, science is hard. Science is also exciting, but the biology and the organisms themselves are going to keep you going late at night when you're locked away in the Farlow trying to finish your dissertation. Yeah.
That's great. Thank you, Ben.
Of course.
It's a pleasure.
Thank you. Yeah.